Okay, can someone confirm that you can hear me?
Yes.
All right, so the headphone doesn't work, so I'm going to go ahead and get started. Didn't help. There we go. So today is February 24th, and we're supposed to be on chapter 14. We're supposed to finish it today. We're a little behind schedule, so I don't know if we will finish chapter 14 today. There is no lab today. You guys should be working on your unknown project. A large number of you have not started that project, and I'm not sure you will be able to finish the project in time now. You are beginning to run out of time to work on your unknown project. Any questions about any of that? Let me share my screen. You guys should have mentioned I was showing you my syllabus and the schedule, and you weren't seeing the screen. Fern, you're seeing my screen now?
Yes, we are.
Thank you. All right, last time we had talked about the human microbiota, and we had discussed this slide saying where the normal microbiota were found in the human body and where they're not, and then the difference between the normal microbiota and the transient microbiota. Questions about the microbiota? When we're talking about the normal microbiota, it establishes a relationship with the human host or its host. When we look at a human, we find that humans have 1 times 10 to the 13th human body cells. And if we look at the normal microbiota in a human, we find 1 times 10 to the 14th bacteria cells. And that begs the ask the question, are we more microbe than we are human? And don't let that freak you out too much. Some pictures of the normal microbiota, the orange cocci in the nasal epithelium, bacteria in the stomach, that's the wall of the stomach, bacteria in the large intestine, you're seeing rods there, cocci or cocci bacillus there, cocci bacillus there. And that I assume is food product. Being digested, of course. There's been some recent surprises in the study of the human microbiota. They've learned fairly recently in the last, well, within the last 20 years, it's about, what is that, 15 years ago, something like that, a little over 15 years ago. That's over 17 years ago, where they found that urine in the bladder, at least the female bladder, is not sterile. There is bacteria, that's normal microbiota, growing in the human female bladder. And they didn't look at males. I'm not sure if that's because males have a longer urethra, and so maybe the bacteria don't get up into the bladder. But in the female bladder, there are bacteria growing in it. There's also bacteria in the human breast tissue, so that mother's milk is not sterile. When the baby is nursing from the mother, it can get mother's milk, but it'll get bacteria in the mother's milk. Any question about that? And I'm not sure why this is a big surprise, but they found a normal microbiota in the placenta. Why this is not a surprise to me is that the human female reproductive system is open to the outside, and the placenta isn't that far in. And there are normal microbiota that colonize the placenta. If you don't know, the placenta is only present when there's a fetus in the mother. The relationship between the normal microbiota and the host is one that benefits both the normal microbiota and, well, certainly benefits the normal microbiota. It can benefit the host as well. For the host, the normal microbiota can engage in microbial antagonism, where the normal microbiota competes with a human pathogen. Some examples where the normal microbiota prevents the growth of a human pathogen by occupying the niche that the pathogen may occupy, like my skin. Hopefully you can see this. Is coated in a normal microbiota. And if a human pathogen were to fall on my skin, it's likely to be around or on top of a normal microbiota cell, in which case the pathogen may not get established because it doesn't have any space. They can compete for resources, like the normal microbiota of my skin will be using the resources there, and then the human pathogen cannot obtain resources, and then the cell would die. The normal microbiota can produce acids. This is particularly common in the normal microbiota in the mouth, in the urethra, and on the skin, where they produce acids, and then the environment is not favorable, it's too acidic for the human pathogen to grow. Some normal microbiota can produce bacteriocins. This is a protein that the normal microbiota makes, and it works like an antibiotic in that it prevents other bacteria from growing. Now, the difference between a bacteriocin and an antibiotic is an antibiotic works on a large number of microorganisms. A bacteriocin only works on bacteria similar to the bacteria that made the bacteriocin. Does that make sense? For example, if we have E. coli in our gut producing a bacteriocin, which your E. coli in your gut do this, the bacteriocin will prevent the growth of Salmonella and Shigella, but it will not prevent the growth of other microorganisms. Any question about any of that? So a bacteriocin is very specific on what it harms, but it acts like an antibiotic. And I read a paper, it's now several years, probably over 5 years ago, where I read that they're using bacteriocins experimentally on patients. And I always wondered why they weren't using bacteriocins on a patient, because if you get like a salmonella infection, you would like to use a bacteriocin that prevented salmonella from growing, but you don't want to prevent other bacteria from growing, such as, oh gosh, what is it called? Fecalis, what is it? Other bacteria in your gut, is it no, I don't remember the name of that now. Enterobacter fecalis, that doesn't sound right. Something like that. And other gut bacteria, such as E. coli. Any question about any of that? Okay. Probiotics are when you consume live microbes that are applied or ingested to the body, and they're intended to exert a beneficial effect. Like this picture here, somebody's eating yogurt that has live microbes in the yogurt, and you can get probiotics that way. Yogurt is actually a good thing to eat, especially for females, because the bacteria in the yogurt can help prevent a woman from getting a urethral yeast infection. Any questions about any of that? Everyone good? Can anyone confirm that you're good?
Yeah, we also tend to give probiotics to people with C. diff.
Right. Oops, don't want to do that. The trouble with C. diff is it makes, just a minute, I'm drawing a blank here. I'm not doing too well today. It makes endospores. And when you treat with antibiotics, that will kill the vegetative cells, but not the endospores. And so when you're treating for C. diff, you have to treat the first time and then expect that it'll come back. Although if you add probiotics, it might compete with the C. diff. And then when it comes back, you let it sit for a little while so that the endospores all germinate, and then you treat with antibiotics again, and then you can usually remove the C. diff the second time you treat. The human normal microbiota forms a relationship with the host. This relationship is symbiosis, where two different organisms or two different populations live together. The normal microbiota and the host engage in a specific symbiotic relationship, which is either mutualism or commensalism. Symbiosis is all organisms living together. Mutualism is where both organisms benefit, like Staphylococcus epidermidis on my skin benefits both the host and the organism. I provide a home for the bacteria. I provide food for the bacteria. And then the bacteria help prevent a human pathogen from getting established on the skin, as the Staphylococcus epidermidis competes with the human pathogen and also makes the skin more acidic. So the human pathogen may not be able to grow in the acid environment of the skin. Another example of mutualism, which is even better, is E. coli in my gut. I provide the E. coli a home, my gut. I provide the E. coli food in my gut, which the E. coli digests. And then the E. coli helps me digest the food, as well as potentially competing with other bacteria, including human pathogens. So the E. coli benefits me in two ways. Any question about that? Okay, that's mutualism, where both organisms benefit. Commensalism is where one organism benefits and the other organism is unaffected. A good example of commensalism with humans is where there's a nematode in our eye. So each of our eyes has about one nematode in it, and it must eat something that falls into our eyes or bacteria in our eyes, because it does not damage us, doesn't eat our cells or even our dead cells. And I don't know what it eats, but I'm guessing it eats the bacteria that falls into our eyes. We provide a home for the bacteria, and then not the bacteria, we provide a home for that nematode, and that's about it. and then the nematode does not harm us. So that's a commensalistic relationship. I could talk about the bacteria on our skin, but like I said, if it competes with a human pathogen, that is benefiting us. Even if it's a slight benefit, that would be mutualism, not commensalism. Parasitism is where one organism benefits. It gets a home, it might get food from living on the human, and then the other uh, loses out. It doesn't benefit from the relationship. Like when a human pathogen gets on us and we give it a home and it, I don't know, might lyse our cells, getting nutrients from our cells, but we get no benefit and it does harm to us. Any question about symbiosis? Okay, the normal microbiota does not normally cause disease in healthy individuals, but sometimes it can cause disease when it hits or is growing on an immunocompromised patient, or when it's growing in a location it's normally not found, in which case it can be an opportunistic microorganism where it's a normal microbiota, but it can cause disease under certain circumstances, like a patient who's immunocompromised. Examples of opportunistic pathogens is E. coli when it's growing outside of the gut. It's normally found in the gut and it doesn't cause any problems in the gut. And we're talking about normal E. coli. But if it's growing in the ear, it can cause an ear infection because E. coli is not normally found in the ear. Staphylococcus aureus is normally found in the nose and the throat of about 50% of Americans. And on 20% of Americans, you can find it on their skin. The trouble is Staphylococcus aureus can cause skin infections, so we call this organism a human pathogen. Any questions about any of that? All right, let's turn to the etiology of infectious diseases. Koch postulates is something you use to prove that an infectious agent causes a specific disease. And Robert Koch was the first person who did this, where he proved that a bacteria, Bacillus anthracis, caused the first bacterial disease that we identified, that was anthrax. And he wrote up his procedure for how he showed that Bacillus anthracis causes anthrax, and that became known as Koch's postulates, the steps you use to show that a given microorganism causes a given disease. I can't blow that up that way. Oh, yes, I can. So you have a sick animal, like this bunny here, and you isolate the pathogen out of the bunny. You grow up the organism in tissue culture, isolate it, get individual colonies, grow up the individual colonies to get pure cultures, and then inject the pure culture into a healthy animal. If that healthy animal gets sick, the same disease as before, then we can say that this organism, which would be one colony here, caused this disease. And why the bunny is on its side is because it's sick, the poor bunny. Koch's postulate says that's not enough. You need to show that the organism in this sick bunny with the same organism you grew. So you got to obtain the organism, the pathogen, from this sick bunny that you injected into the bunny to make it sick. And if you can do all that, then you can show that this organism causes this disease. Any question about that? Well, there should be a question because I haven't talked about the fifth part of Koch's postulates. And the other thing is this organism must be present in every case of the disease. Like you cannot have an individual who's sick with this disease and it's not caused by that agent. For example, pneumonia is caused by many different agents. So you can't use Koch's postulates to go on and prove that Klebsiella pneumoniae causes pneumonia because there are other agents that cause pneumonia. Any question about any of that? This is the second time we've talked about Koch's postulates. It might even be the third time we've talked about Koch's postulates. We're going to talk about Koch's postulates 3 times in this course, and it's important enough that we're going to talk about it 3 times, and there will be at least one question on the final on Koch's postulates. And I think you've already had one question on Koch's postulates on a quiz, and there could be another question on Koch's postulates on a future quiz, but certainly the final. Any question about any of that? Now, Koch's postulate is the gold standard for showing that a given microorganism causes a given disease. But there are exceptions to Koch's postulates. Like what happens if the microbe has unique culture requirements and cannot be cultured on artificial media? For example, the disease causing syphilis, meaning the organism causing syphilis, which it's escaping me now. Like I said, my mind isn't working well today. So if anyone knows off the top of your head, you can state it. I almost had it, but we'll move on. This disease cannot be grown in tissue culture. And they've been trying to grow it in tissue culture for over 100 years. They've never figured out what this organism that causes syphilis requires. So they've never been able to find the right additives to get it to grow. And yes, they've tried growing it on blood agar plates. It doesn't grow.
I'm not going to butcher this, but it's Tremponium something.
Yes.
Pallidum.
Pallidum, yeah.
Yeah.
Treponema, there it is. Treponema pallidum. You actually say the pallidum twice because there's different sibling species for that one. But thank you. Another one is Bartonella henseli. I remember that one. It causes cat scratch fever, and they've never been able to get that to grow in tissue culture. So you can't use Koch's postulates to show that these 2 organisms cause a specific disease, either syphilis or cat scratch fever, because you can't get it to grow in tissue culture. And then what do you do if multiple organisms cause the same disease? Meaning the patient has the same signs and symptoms, and it could be pneumonia. There's multiple agents causing pneumonia, or even the human cold. There's many species of viruses that cause the human cold, and they come from 3 different families of viruses. So you can't prove that these cause the given disease using Koch's postulates. And then some species can cause different disease conditions, like salmonella, which that's not the species name. What the heck is the species name? I'm not remembering here. Salmonella, you know what bacteria I'm referring to. Is it Salmonella enterolytica, something like that?
Is it the
Uh, oh, yeah,
Typhiri?
Typheria is one of them. There's about 3
Mm.
Salmonella that cause disease. Um, anyway, Salmonella can cause more than one disease. It can cause salmonellosis, which is not that severe of a disease. The patient will have diarrhea, usually for just a couple of days, and then they'll get better. And then salmonella can also cause typhoid fever, where the patient is usually sick for like 2 weeks or more, and they're severely sick. All of these diseases, you can't use Koch's postulates to prove that this organism causes the disease. And then what can you do when there's moral and ethical conditions for a human only disease? For example, HIV, that agent, has never been injected into people to look and see whether HIV causes AIDS, because it would be unethical as well as moral implications for doing an experiment like that. And how then do we prove that HIV causes AIDS? Anyone have a guess?
Didn't they use monkeys?
Uh, HIV doesn't grow in monkeys, it does grow in chimps. However, chimpanzees do not get AIDS.
Oh,
So
that's interesting.
they do get an infection of HIV, but it's not a lethal infection and they do not get AIDS,
Hmm.
but it will grow in them. Um, no, that's not how they did it. What they did was they followed large number of people using an epidemiological study over time. And then they just followed people who were HIV positive, and some of them developed into AIDS. And probably, if you follow them long enough, all of them will probably develop into AIDS. Um, and then they also followed people over time, and some of them were HIV negative, and they became HIV positive. And then they went and followed them over time, and some of them became AIDS patients. So we do know that HIV does cause AIDS, but we do not know it from Koch's postulates because no one's ever injected HIV into people and then they developed AIDS. Any question about any of that? All right. Classifying infectious diseases, there are different ways to classify them. A symptom is a change in a body function that is experienced by a person as a result of a disease. But this is a subjective, I guess we'll call it symptom, in that there's no way to measure the symptom. Like if a patient has a headache, there's no way that anyone else can measure or verify that the patient has a headache. You just have to rely on the patient telling you. And if the patient can't communicate, boy, you're out of luck on that one. Um, a sign is a change in the body that can be measured or observed as a result of disease. For example, when someone has a temperature, you can measure that temperature using a thermometer and say they have a fever of 101 or whatever. A syndrome is a specific group of signs and symptoms that characterize the disease, such as the AIDS syndrome is characteristic of certain signs and symptoms that are characteristic of a patient having AIDS. Any question about any of that? Now with this, symptom and signs, this is a textbook definition, although you could run into these terms in the medical profession. But you should realize with most patients, they will use the two words synonymously. But a symptom is one that the patient can feel, but nobody else can measure. A sign is something the patient can feel, but you can measure. A communicable disease is any disease that can spread from one host to another. Some examples of a communicable disease include measles, tuberculosis, called TB, chickenpox, communicable because when one patient gets it, they can spread it to another. A contagious disease is a disease that is easily spread from one patient to another. So a contagious disease would be COVID-19, the human cold, or the flu, a disease that easily spreads from one patient to another. All contagious diseases are communicable diseases, but not all communicable diseases are contagious diseases. So measles and chickenpox are both communicable and contagious, but tuberculosis is communicable, but it is not contagious because it does not easily spread from one host to another. Any question about any of that? A non-communicable disease is a disease that does not spread from one host to another. For example, when a patient gets tetanus, they are unlikely to spread tetanus to another patient. And there's only one example that I can think of where tetanus could actually spread from one patient to another. Can anyone guess how that could spread? How tetanus could spread from one patient to another?
Does it have to do with bodily fluids?
No, tetanus doesn't spread through bodily fluids. Tetanus usually is obtained in something like a puncture wound. So that's why if you step on a nail or something like that, you might go get a tetanus shot, which probably it's due for me to get a tetanus shot. No, the only example that I can think of where tetanus would spread from one patient to another is among cannibalism. If you were to eat a patient who had tetanus, you could come down with tetanus. But other than that, tetanus does not spread from one patient to another. Any question about communicable diseases?
I have a question. So did the microbiology department decide to only do Zoom because of the variants of COVID?
When COVID first came out, Clark College closed and we were going to switch to online. But microbiology is only online at Clark even now because of the variant coming around, which is the latest one. What the heck was it?
Omicron.
Micron, yes.
Now there's another one.
Is there another one that's spreading? I haven't heard about that.
Yeah, now it's 30 times more contagious than the last.
I thought it's not Micron. What the heck is it?
It's
But
Omicron.
Omicron, there we go. Omicron. I thought it was 30 times more contagious.
That's what they said, but I do a call with the Department of Health for my work, and we just heard that there's another one that was found.
Well, that's not good. I'm not surprised, but I was hoping that COVID-19 would decrease before the next variant came out. But it sounds like the next variant may be already on the rise, in which case it looks like we're going to be with COVID-19 with more and more variants always coming. And we will see what happens. The nice thing about Omicron is that it was much less lethal than most of the other COVID-19 variants, even though it was derived from Delta, which was the most lethal of the COVID-19 variants. And they expected that the next variant would be related to Omicron. And so they were hoping it would be less lethal. Obviously, we wouldn't know right now what its characteristics are because it just was started. And I think they're still working on the characteristics of Omicron. One bad thing about Omicron for you guys is that COVID-19 was not hitting young people very much, and Omicron and Delta were tending to hit young people more often than the other COVID-19 variants. Any question about any of that?
No.
We will talk more about COVID-19 later in the term. If there's no questions, let's move on. Shrink that. The occurrence of disease. To understand the full scope of disease, we need to know the details about its occurrence. When we're talking about the occurrence, we're talking about old and new cases. Like usually in the United States, when we're talking about COVID-19, we're talking about both old and new cases. The incidence of a disease is the fraction of a population that contracts a disease during a specific time, usually the current time. And this incidence is talking about new cases only. On the other hand, the prevalence of disease is the fraction of the population having a specific disease at a given time. So in 2007, when talking about AIDS in the United States, the incidence of AIDS was 56,300. And these were the people who came down with AIDS in the last year. So only the new cases. The prevalence of AIDS in 2007 was 1,185,000. And that's both old and new cases as of 2007. And if you don't know, the prevalence of COVID-19 is getting close to the number of AIDS cases in 2007. Meaning, I believe it's over 1 million people now in the United States who have come down with COVID-19. And that might be, yeah, it's 900 million people. That's not actually the prevalence of people who came down with COVID-19. That's about the number of people who've died of COVID-19, because I know the number is over 900 million now. All right. Sporadic disease is a disease that occurs only occasionally in a population. For example, we can say typhoid fever is a sporadic disease in the United States. Another good example is malaria. is a sporadic disease in the United States. And how we get malaria is people travel outside of the United States, pick up malaria outside of the United States, and then return to the United States, and then malaria develops in them. Anyways, an example of a sporadic disease. An endemic disease is a disease that is constantly present in a population. For example, in the United States, the common human cold is endemic, and it would be true for any place in the world. Malaria is endemic to the tropics, and it's not found normally in the United States. An epidemic is a disease that's acquired by many hosts in a given area in a short time. For example, when we have an epidemic of influenza, and usually they say an epidemic is where the number is significantly greater than normally found. A pandemic is when you have an epidemic, which an epidemic, I should state, is on one continent. If it spreads to 2 or more continents, we then call it a pandemic. So a pandemic is a worldwide epidemic. An example of a current pandemic is COVID-19. We're under a pandemic of COVID-19 because before 2 years ago, there was no COVID-19 cases in the human population. Any question about that? When we're talking about disease, it's sometimes important to talk about the severity or duration of a disease. This is a useful way of defining the scope of a disease in its terms of severity or duration. An acute disease is where the symptoms develop rapidly, but the disease is short-lived. For example, the human cold is an acute disease. The flu, even if it's severe, is usually an acute disease because the symptoms develop rapidly and it's relatively short-lived. You will have it maybe for a month, but very few cases of the flu go beyond a month. And usually it's only about a week or two. A chronic disease is a disease that develops slowly and persists for long periods of time. For example, mononucleosis. The patient gets infected, they get sicker and sicker, and then they're in a constantly, constantly run down state for several months. And another example would be hepatitis B, causing liver damage, chronic disease. Now, there are diseases in between an acute and a chronic disease, and the name for that is a subacute disease. But I'm not going to test you on this because I don't think anyone is familiar with subacute sclerosing panencephalitis, but it just has symptoms in between a chronic disease and acute disease. Another case of a chronic disease is somebody who plays tennis a lot and the tennis elbow starts developing. And that's usually a chronic disease. As long as they continue playing tennis, they'll continue having chronic tennis elbow. And usually you will have tennis elbow for the rest of your life, even if you stop playing tennis. Meaning the damage was done, and then you're going to have to live with it. Any question about acute diseases and chronic diseases? If not, let's move on to the extent of host involvement. Infections can be classified according to the extent to which the host body is affected. There are local infections where the pathogen is limited to a small area of the body. For example, a boil or an abscess of the skin is a local infection caused usually by Staphylococcus aureus. But that infection can spread, and if it gets into the blood or it's spread throughout the body, we call it a systemic infection or a generalized infection, an infection throughout the body. As I mentioned, the boil caused by Staphylococcus aureus could get worse and worse, and then it could infect the blood. The blood can then carry it, the Staphylococcus aureus, throughout the body, and then we get a systemic infection where many tissues and organs could be infected. Another example of a systemic infection is when a child or a patient comes down with the measles. They'll breathe it in, it'll be in the blood, it'll be carried to the blood to other body tissues. And then it'll reproduce, it'll go into the blood again, and then it'll be breathed out, and then somebody else can get the infection. The point is, it's a systemic infection. There are focal infections. This is a systemic infection that it began as a local infection. The perfect example of that is Staphylococcus aureus causing a boil, or MRSA causing a skin infection. MRSA is a multi-drug resistant Staphylococcus aureus, and it spreads from the skin into the blood, and then will be carried by the blood to other places in the body. All right, any question about any of these? Bacteremia is when we have the presence of bacteria in the blood. So when you look at under a microscope and you see bacteria in the blood, we call it bacteremia. Septicemia is similar, but this is when the bacteria are growing in the blood. And you'd have to have a specific test to show that the bacteria is growing in the blood. Usually the patient who has a blood infection, it doesn't matter, bacteremia or septicemia, because it's in the blood, that's bad. And usually it'll grow in the blood. Toxemia, the presence of toxins in the blood. Viremia, the presence of viruses in the blood. The state of host resistance also determines the extent of an infection. A primary infection is an acute infection that causes the initial illness. Like somebody gets a sore throat, that's the primary infection. Then the sore throat does damage to the throat, maybe damage to the ear, and then that can lead to secondary infection, where an opportunistic organism infects the patient after the primary infection has weakened the patient and done damage. So now the secondary infection or secondary agent can move in and colonize the area. Any questions about a secondary infection? A subclinical disease is also called an asymptomatic disease, is where the patient has the infection, they have the disease-causing organism, but they have no noticeable signs or symptoms. And if young people who have COVID-19 about 50% of them are subclinical. They have the disease-causing agent, and they can spread the disease-causing agent, but they have no signs or symptoms of COVID-19. Actually, some people can carry the polio virus and never get polio. I was just reading about a case of polio in Africa. I don't remember the country, but apparently was brought into Africa by a subclinical patient. And then that patient spread it to somebody else in Africa, and they came down with polio. The first case of polio virus in the continent of Africa in about 10 years. Any case questions about that? If not, let me mention the polio virus is normally only found in 2 countries in the world, and that's Afghanistan and Pakistan. All other countries of the world, except for that one country in Africa now, are polio free and have been for at least 5 years. The United States has been polio free for over 20 years.
Is that because of like lack of access to the vaccine?
Well, the trouble in Afghanistan was there was so much fighting going around that it was really hard to inoculate the population. And in Pakistan, there's not as much fighting going around, but it's sort of like guerrilla warfare. And that region of the world, Pakistan and Afghanistan, are next to each other. They don't want to receive the vaccine, so they're resisting getting the vaccine. And so that's why this region of the world is the only 2 places where you can find polio.
Hmm.
Okay. The World Health Organization is trying to make polio an extinct disease, and they've succeeded everywhere except for those 2 countries. Now, the first world countries got rid of polio a long time ago on their own. And then there was one doctor who had the brilliant idea of getting rid of polio in South America. And he succeeded. And after he succeeded, the World Health Organization said, we should get rid of polio in the world. And so they've been working on it for over 20 years now. And they got rid of polio in everywhere but 2 countries. And like I said, it's going to be hard getting rid of it in those 2 countries, although they're more stable now. There's less fighting in Afghanistan and Pakistan than even just a year ago. But like I said, the people don't really want the vaccine, and you do need enough people vaccinated to keep the polio from spreading. All right, so let's talk about the severity or duration of disease. A latent disease is a disease where the disease-causing organism is present, but the agent goes dormant or something similar to dormant and does not cause disease. So the patient has no signs or symptoms of disease. The pathogen is said to be inactive or latent. The classic case of that would be shingles. A child normally comes down with chickenpox, and then the disease virus is active, replicating while the child has chickenpox, and then the chickenpox goes away, but the disease-causing agent does not. The disease-causing agent goes latent, and this latency could last for decades. And then the agent, the virus becomes active and causes disease, and the patient can spread the virus. And when the patient gets disease, it's no longer called chickenpox, because the second time you get it and all later times you get it, the disease is called shingles. And the signs and symptoms of chickenpox and shingles differ. So this is definitely a one agent that causes two diseases. Chickenpox and shingles are definitely different signs and symptoms. And in between, the patient getting chickenpox, usually when they're a child, and then the patient getting shingles, usually when they're elderly, can be decades. Any question about any of that? Another good example of a latent disease is the herpes virus that causes cold sores. When it is active, you have a little bump and it's usually sore, and the virus is growing in that bump and it can spread. And then it goes latent. You cannot spread the disease. The agent is latent, not replicating, just hanging out. And then a year or so later, you come down with another cold and you get a herpes sore again, which we call a cold sore. Any question about any of that? All right, let's talk about herd immunity. If you have many immune people in the population, they can prevent the spread of an infectious disease. Let me blow this up. With COVID-19, when it first hit the human population, everybody was susceptible. A few people first got it, and then everyone in contact with them got it, and they were in contact with more people. And then eventually, if there would not have been controls in place, the majority of the population would have come down with COVID-19 infection. If, on the other hand, most of the population are immunized, the yellow individuals, and then the red individuals are the contagious people, only someone who is not immunized and in contact with the COVID-19 infected patient could get the disease. So this individual here would get the disease, and this individual here would get the disease, but the disease can't spread to the immunized individuals, and so the disease doesn't spread further. And you have to have enough of the population immunized to prevent the spread of disease. You don't have to have all the population immunized, but with COVID-19, they know, or knew, I should say, that you had to have about 85% of the population immunized to prevent the spread of COVID-19. With COVID-19, a little over 50% of the US population is immunized. And like I said, you need 85% of the population to be immunized to prevent the spread of COVID-19. And that's not counting the new variants like Omicron can infect individuals who were immunized with COVID-19, and they can spread the disease, come down with it, disease, and then spread it to other people, including more immunized people. And the reason is that the Omicron variant of COVID-19 differs enough so that the vaccine is not 100% effective. And so you can get infected, and you can come down with disease, even though
Is
you are immunized.
is that because of the different protein spikes on it, like on the different variants?
It's because not the protein spike, but there are different proteins, and they may be just different in one amino acid, but the antibodies to COVID-19 can't recognize that protein, and so those antibodies don't bind to it. And it's just that Omicron is different enough that the vaccine isn't providing close to 100% protection, and they don't know what the protection is of Omicron. I've never read that they've figured out what it is yet, but for Delta variant, it only, well, the vaccine only protected you 65% of the time, and that's down from 95% of the time. And Omicron was protecting you, or the vaccine was protecting you less than 65%. And I don't know what it is. My guess is it's like 50% or a little bit lower. And it's just that the variant is different enough that the vaccine is not offering the protection we normally associate with a vaccine. And they are working on a vaccine for Omicron, but the problem is before that vaccine dose will become available, Omicron will probably go away by that time. Any question about any of that?
It's interesting because we had people who were vaccinated and boosted who got the last variant but had no symptoms.
Yeah, that's one benefit of getting the vaccine, and that is the symptoms will be much milder if you had the vaccine than if you did not have the vaccine. And the number of deaths in a vaccinated individual is much less than the number of deaths in the unvaccinated. Anyways, in the United States, we'll have problems getting enough people immunized to COVID-19 to prevent its spread. Although, if you put in the population that have been infected by COVID-19 with the population that is immunized, we may get to 85% of the population being not vaccinated, but vaccinated or immune because they got COVID-19.
Oh, like developing their own antibodies.
So they're developing their own antibodies, but the real problem with COVID-19 is there will probably always be new variants, and the new variant will be more likely to get through the vaccine. And that's the other problem we'll have with COVID-19. I suspect that with COVID-19, we'll just always have a new variant coming out, and unfortunately, we'll never go away from COVID-19.
So it's kind of like how the Spanish flu started, and now there's different variants of influenza.
Kind of. All right, let's move on. Patterns of disease. Normally, there are specific sequence of events associated with an infection by a microbe. And then there's a specific sequence where there's a subsequent disease forming. A reservoir must exist for the disease-causing agent. Before someone can become infected, they have to get the disease from a reservoir. The pathogen must be transmitted to a susceptible host. The pathogen invades the host and multiplies in the host. That's why the patient gets an infection from the invasion and multiplication of the agent and the host. The pathogen must, on top of that, injure the host. You would have no signs or symptoms of disease if the pathogen did not injure the host. And maybe that's what's going on in the case of COVID-19 when we have young people who are infected with the agent, the virus, but they're having no signs or symptoms. And that is, they're not being injured. Not enough cells are dying so that there's damage to the patient. And then the illness ends in one of two ways. One, the host immune system ramps up and destroys the pathogen, and then you're disease free. Or two, the illness can end when the patient dies. Very different ways that the disease can end, but those are the two ways that disease can end. And these patterns of disease always occur, and usually in this order. The reservoir exists, and from the reservoir, the patient gets infected, being transmitted to the patient. The pathogen invades the host and then multiplies in the host. If the patient gets the disease, the pathogen injures the host. And then usually the host immune system destroys the pathogen, but the patient could die, and that will end the illness. There are predisposing factors that make a patient more susceptible to getting a disease. For example, females have a shorter urethra, so they have a higher incidence of a bladder infection than males. There are some inherited traits that make a patient more susceptible to disease, like sickle cell anemia. The sickle cell gene makes the patient much more susceptible to other diseases. This isn't exactly an inherited trait, but diabetics are more susceptible to getting disease than other patients. Climate and weather can make a patient more susceptible to getting disease or predisposing the patient to getting disease. We all know that colds and flus are more common in the wintertime. And it's more than just that we're all indoors, breathing the same air. That makes the disease more likely to spread. But it's also true, they've discovered that colds and flus can live longer in the cooler weather of the winter, and then they live longer in the sun of the winter because the UV light of the sun is weaker than the UV light of the summer. So the point is climate and weather are predisposing factors to disease. Fatigue is a predisposing factor to disease. As you know, you're a student, and students often don't get enough sleep because they're studying or out partying, and they become fatigued, and that makes them more likely to come down with disease. Age is a predisposing factor for getting the disease. The older you are, the more predisposed you are to getting an infection. And then usually, if you get the infection, the older you are, the more severe the disease will be. Lifestyle can infect you're getting a disease. For example, smokers are much more likely to come down with a respiratory disease than non-smokers. And it's because smoking damages the respiratory system. And if the respiratory system is damaged, the invading pathogen has an easier time invading. Chemotherapy can predispose a patient to getting disease. Generally, chemotherapy, not only can it damage the the body's defenses, but it weakens the immune system. And so you're more likely to come down with disease or a serious disease if you're taking chemotherapy. Any question about any of that? Disease tends to develop in the same way. Once the host defenses are breached, disease develops following a certain sequence. And this sequence tends to be similar for both acute and chronic diseases. When you first become infected, till when you first have signs or symptoms of disease, it's called the incubation period. So on the y-axis, we have the number of microbes, as well as the intensity of the disease. And on the x-axis, we have the time. So when you're first infected with disease, till when you first have signs or symptoms, that's called the incubation period. The disease is growing in you, but it's the disease-causing organism are not high enough to cause disease, signs and symptoms. When you first have signs and symptoms, and the signs and symptoms are mild, we call that the prodromal period. For example, you're going around and then you say, geez, do I have a sore throat? And you have to think about it. And you say, yeah, I think I'm coming down with something. I have a slight sore throat. That's the prodromal period, where the signs and symptoms are mild. And then the signs and symptoms increase, the severity of the disease increases, as well as the number of infective agents increase. My mouse died. This is called the period of illness, where the patient has the most severe signs and symptoms. Like you don't have to think about whether you have a sore throat or not anymore, because you know you have a sore throat. The patient then gets better. The signs and symptoms start decreasing. Usually, if you have a fever, the fever breaks. There's a term for that. I forgot what it's called when the fever breaks and the patient starts getting better. That's called the period of decline. And then after the number of organisms in the patient drop below a certain threshold, we enter the period of convalescence, where the patient has no signs or symptoms of disease, but they still have the disease-causing agent. My mice died, there it is. So you still have the disease-causing agent, but you have no signs or symptoms of an illness. These signs and symptoms follow most diseases. Now, with that stated, you may not have certain signs and symptoms. Like, for example, when I've had the flu, sometimes I'm walking around and everything's fine. I'm in the incubation period. I don't even realize I'm sick. And then the next moment, it feels like somebody hit me over the head. I have a very intense headache. And it feels like they hit me over the head with a sledgehammer. So there is no prodromal period. You go immediately from incubation to the period of illness. And then if you die in the period of illness, you have no period of decline or period of convalescence. HIV in the past used to not have a period of decline or period of convalescence because the patient would die. And that could happen today. It's just that it's less often than it was in the past because of new medications and keep the patient alive. Any questions about any of that? If not, here's a question for you. During what period are you not likely, and I'm going to just say, are you not able to spread disease from yourself to another individual? At what period are you not communicable?
The decline?
The decline period. Anyone else? I'll give you a hint. It's a trick question.
When you have the illness?
You see that, no, that's really a trick question. You have the disease causing agent in the incubation period, the prodromal period, the period of illness, the period of decline, and the period of convalescence. So in all of these periods, you can spread disease.
Oh.
Well, that's why it's a trick question. My mother would always tell me when she was sick, oh, I'm getting better now, so I can't spread disease. And she would hate that I'm telling you. And I'd say, Mother, I'm a microbiologist. I know better. In the period of convalescence, as well as the period of decline, you can spread the disease. You can spread the disease in all of the periods, not until you're after the period of convalescence and you have no infective agents, will you not have the capability of spreading disease. For common illnesses like colds and flus of adults, and this is only true of adults, for colds and flus and common illnesses, you're most contagious late in the incubation period and in the prodromal period. Why you're more contagious in these periods than in the period of illness is with adults, your immune system is acting. And because your immune system is acting, you're less likely to spread disease in the period of illness for the colds and flus than you are late in the incubation period and in the prodromal period. Okay? And that's true for adults. That's not true for children for colds and flus. They're likely to spread a cold or flu in the incubation period, late in the incubation period, in the prodromal period, as well as the period of illness. And you don't need to know the difference between children and adults, but you should know about adults. That you're most contagious late in incubation and the prodromal period. And that's probably true for COVID-19 as well, but I don't think anyone's ever studied it for COVID-19. All right, spread of infection. There are reservoirs that are a source of infection. They are continual sources of disease organisms. Reservoirs can be living or non-living. There are three main reservoirs. Human reservoirs. For all human diseases, humans can be a reservoir. For many human diseases, humans are the only reservoir. Like AIDS, we can only get this from another HIV infected patient. Gonorrhea, also humans are the only reservoir. Polio, humans are the only reservoir. Carriers may be inapparent infections, meaning the patient could be asymptomatic or they could be in latent disease. Although usually a latent disease, you would not spread the disease. But an asymptomatic patient can spread the disease. There are animal reservoirs. Like rabies, humans can only get rabies from an animal. Lyme disease comes from an animal reservoir. Many diseases that we get from animals we call zoonoses, where they're an animal disease, but they may be transmitted to humans, like rabies and Lyme disease. Rabies does not spread human to human, so humans can only get it from another animal. There are also non-living reservoirs. For example, botulism and tetanus, we get that from non-living reservoirs. Botulism, usually you get it from canned food. Tetanus, usually from a puncture wound. Non-living reservoirs actually are very common, where we get the disease from the soil, the water, or from the uncooked food we eat, such as lettuce. Any question about any of that? If not, I'll stop here and I'll see you at no, I'll be at the lab at 6:30 to 6:45 to answer questions. Okay?
Thank you. Have a good night.
All right, you too.
Thanks.